Molecular cloning is a method in molecular biology where scientists copy a specific piece of DNA and insert it into another DNA molecule so that it can be stored, studied, or used by living cells. It allows researchers to isolate one gene from a large genome and make many identical copies of it, much like taking a single chapter from a very large book and photocopying it so you can study it more easily. In simple terms, molecular cloning is the process of copying a gene and placing it into a new biological context so that it can be analyzed or used. Why molecular cloning matters? DNA inside cells contains thousands of genes. Studying one specific gene directly inside a cell can be difficult because it is surrounded by so much other information. Molecular cloning allows scientists to extract just the gene of interest and work with it in a cleaner, controlled environment. This is essential for understanding what genes do, producing medicines and vaccines, engineering bacteria to make useful proteins like insulin, researching genetic diseases, advancing biotechnology, and synthetic biology. A helpful analogy. Think of DNA like a library full of books. Each book is a chromosome, and each chapter is a gene. If you want to study one chapter closely, you find the chapter, make a copy of it, place the copy into a clean notebook, give the notebook to a cell that can store it, and make more copies. This is exactly what molecular cloning achieves at the cellular level. Key components involved: gene of interest, the DNA segment you want to study or use. Vector, a small, circular piece of DNA, often a plasmid, used as a carrier for the gene. Host cell, usually bacteria like E. coli that replicate the plasmid and produce copies of the gene. What is a plasmid? A plasmid is a naturally occurring, circular DNA molecule in bacteria. Scientists use plasmids because they are easy to manipulate, copy themselves inside cells, can carry added genes without harming the host. Conceptual overview of the cloning process. Identify the gene you want to copy. Insert the gene into a plasmid. Introduce the plasmid into a host cell. Let the host cell grow and replicate, making many copies of the plasmid and the gene within it. Collect and analyze the copies of the gene or the protein it produces. The key idea: the host cells act as biological factories, making millions of copies of the gene for you. What molecular cloning allows scientists to do? 1. Study gene function. By cloning a gene and expressing it in cells, researchers observe what proteins it produces and how they behave. 2. Produce useful proteins. Many important medicines are made using cloned genes. For example, human insulin for diabetes is produced by bacteria that contain the cloned human insulin gene. 3. Modify organisms. Cloned genes can be inserted into plants to improve resistance to pests, bacteria to produce biofuels, yeast to make vaccines. This is part of genetic engineering. 4. Explore genetic diseases. Cloning genes with known mutations helps scientists study how diseases develop and how to treat them. Major scientific impact. Molecular cloning is the foundation for gene therapy research, biotechnology companies, modern vaccine development, CRISPR gene editing, synthetic biology. It is one of the fundamental tools that enabled the modern revolution in genetics and personalized medicine. Common misunderstandings. Misunderstanding: it creates whole organisms. Reality: no, cloning a gene is different from cloning an animal. Misunderstanding: it always changes DNA permanently. Reality: often, cloning is just for studying or copying DNA, not altering genomes. Misunderstanding: it requires advanced technology only. Reality: the principles are simple, the impact is enormous. In summary, molecular cloning is a core technique in modern biology that isolates and copies specific genes, inserting them into small DNA molecules so they can be stored, studied, and used by living cells. It allows scientists to understand how genes work and to produce valuable biological products. Put simply, molecular cloning lets scientists take one gene out of a crowded genome, copy it, and use it, like photocopying the instructions for a single component, so it can be studied and applied anywhere.